Hi, everybody. Sorry about that. Just getting set up here. Everybody who is 0 people, great. Okay, well, we're going to get into it. Okay, so today I want to do a little bit of another tutorial on the approach to solving Hardy-Weinberg questions. I'm just getting my iPad going here, and we're going to get that set up. And so I'm going to look up some more Hardy-Weinberg questions right away. And actually, I think we'll just focus on the Hardy-Weinberg questions at the end of that booklet. So we were doing this one. So yesterday we started working on this booklet here, this handout, whatever it is, and I posted this on the classroom. And I'm going to show you a couple of different approaches. One of the approaches we're going to use is this device here to solve some Hardy-Weinberg questions. So I'm going to grab some questions. I'm gonna be able to steal some things here. So let's grab one of these. Okay, now the only thing I don't like about this particular group of questions is that they are, I want to copy that. Okay, delete screenshot. Is that they are a little bit on the wordy side of things, but you know, they do the job. So let's put one of these. There we go. Okay, so this is the question. Scale colorization of lizards has a complete dominance relationship with green scales are dominant over blue scales. There are 1,024 individuals with the genotype of GG, or homozygous dominant genotype. So one of the first things to think about is when they give you a question like that, start building out which of the Hardy-Weinberg equation parts do you have here. So we have this equation, q squared equals 1, and we have p and q. And sort of, you know, one thing to think about is that each of these is their own sort of little section, right? Each of them has their own representation. Here we have p squared, which is representing the homozygous dominant individuals. So that would be this one. So that's going to be my p squared. We have the next one, which, what color did I make them? Oh, red. Okay, so the next one is the PQ. These would be our oops, not them, but our, oh, okay, whoa, wait a second. Oh, how weird. Okay, I see. I got to read these questions carefully. Okay, there are 1,024 individuals with the GG. I thought that was the total. There are 512 with the heterozygous approach, and there is 64 that are in the recessive. So each of those represents a different part of this equation, and knowing with the end result that they have to add up to 1 if we get their fractions. So we want to find the frequency of the dominant and recessive alleles and the frequency of the individuals with dominant heterozygous and recessive genotypes. So this is our dominant, right? Dominant genotype. This is our heterozygote. And this is our recessive, homo-recessive. So when we compare these, when we look at these values, we see that we have numbers, but they are not in a frequency. They're in whole numbers. So to determine a frequency is going to be equal to the number of individuals over the population. And I did see some work the other day that I was looking at for somebody, and I saw that that this calculation wasn't quite happening. So we need to really focus on what does calculation mean. So in this case, we have 1,024, we have 512, and we have 64. All of these have to be added together to find our population. So what's our population? And so the population in this case, let's see if I can maybe a little bit more screen here. So the population is going to be the total 1024 plus the 512 plus the 64. Add those all together, and we get something. Let's see, 8, 9, 10, 10, 6, 7, 8, 8, 9, 10. So it looks like we get 1,600. Does that look right? 1,600. 536. Yeah, so 1,600. So the total is 1,600. So this means that for the GG, which is also the squared, we have 1024 divided by 1,600. And so what do I want to do here? I want a calculator. $10.24 divided by 1,600, 64%, 0.64. Maybe we'll switch pens to match our color notation here. We had G, our heterozygote. This is the 2PQ. And so the 2PQ, there's 512 of those individuals. So that's 512 divided by 1,600. That's 0.32. And then we have an option at the end. We could, for the little guy, the homozygous here, we could calculate it with that 64 over 1,600. Or we know that this total has to add up to 1. So we could also take a subtraction. So let's see what that would look like. We would go 1 minus 0.64 minus 0.32. And we would find that this value should be 0.04. If we did 64 divided by 16, we also get 0.04. So that's confirmed. Okay, so that's how we find the frequency when given the genotype. So somehow, through some sort of biological voodoo here, they were able to measure the genotypes of all their individuals, and they got these frequencies that way. It's not typical. Typically we can only measure the homozygous recessive individuals. But that's what we were able to do that. Now the question they were asking is, how do we get P and Q, which are the probability of the capital G and the probability of the lowercase g, or probability of the dominant, probability of the recessive. And so the answer here is to consider that this equation has only one variable. It has just the p in it. It's p squared. And from math, we can take the square root of a square to determine the value. So the square root of a squared value is the same as the original, as that root value. So in our case, this means that if you put 0.64, which is p squared, take the square root of it, and you will be left with 0.8. And that is our probability of G. Do the same thing with Q. Q is equal to the square root of Q squared. And so that means it's equal to the square root of 0.04. And that means it's 0.2. So this is always the fun part, is that you do all these calculations and then you find out that it's actually a pretty high number, right? 20% of the population is carrying this homozygous recessive gene, or is carrying this recessive gene. But in the visible population, only 4% have that trait. So let's go back and let's do another question that's a little bit more typical. Oh, let's double check. Yep. Okay, we're happy. So let's do the next one. And this one's about the next generation. So this is always the interesting part to go and look at. Is it's one thing to find the equilibrium values of a population, but it's much more interesting to compare that value to the different generation. So I'm just going to do a little bit of shrinking here, so that I can fit in our next question. All right, the next generation of lizards has this many individuals with green scales and this many individuals with blue scales. And they said that the blue scale is the recessive one, right? Green are dominant over blue. Okay, so solve the equilibrium. So we have 108 and 1092. So what we want to do first is find out the probabilities. So, or what's the frequency of recessive type, phenotype. That's sort of our step one. And to do that, we need to take our value, 108, divided by the total number of organisms. So in this case, that would be 108 plus 1092. So we'd be looking at 1,200 individuals. This is our population. Population, type. Divide those away, and we get 108 divided by 1,200. So it's 9%. So already we could say that this is probably a different genotype, right? Or probably different. Look, 0.04, 0.09. So already we know that evolution has occurred. So the population is not in equilibrium. So we can even just do that right away. We can go like this and say, not in equilibrium. Okay. So step 2, we're going to look to solve for Q squared. So one student was suggesting that we could use the Punnett square, which is something they were familiar with. If you're not familiar with the Punnett square, then you don't have to use this mechanism, but it is pretty useful, and we will be using it a lot in the following unit. Okay. So with the Punnett square, we can put our probabilities of P and Q, which we know add up to 1, on top of the square and to the right of the square. And then inside each of the boxes, we can do the multiplication inside this box. Oops. When determining the probability of an event, we always multiply the two probabilities. Or if we were determining the probability of 2 events occurring simultaneously, we have to multiply the events. And so that's why we do a multiplication here. So we have the probability of Q, probability of P, and then in each of the boxes is all the opportunities that are available to us. If you wanted to make an analogy, let's maybe do a little cloud over here. So a cloud would be to say, let's say that we had a gene that worked like a coin, where on one side of the coin you have a face, and on the other side you have a tail, right? So I don't know why. Yeah, it's so good. It's my dog tail. Okay, so we got heads and tails. I don't know why they call it tails when they don't. There's no animals on it. I guess there's an animal on the other side, and all the animals have tails. But okay, so head and tails, and so we know from heads and tails that if you have to, that there's a 50-50 chance. So you could have two heads come up if you flip two coins. You could have a heads and a tail come up. You could have a tails and a heads come up, or you could have two tails come up. This is the same as saying that half of the coins are heads, half of the coin is tails, and we're flipping two coins, half and half. So then you have the probability of two heads, the probability of a heads and a tail, probability of a heads and a tail, probability of a tail and a tail. We can see that every time it's 1/4, which is 1/2 times 1/2 is 1/4. 1/2 times 1/2 is 1/4. 1/2 times 1/2, 1/4. 1/2, 1/2 is 1/4. These two added together become our 2PQ. So that becomes 1/2, because half of our squares. So that's sort of that's an analogous problem that we could do in probability. It's certainly if you're in the applied math stream, you would probably end up doing this quite a bit in the applied course. So let's look at this question now. I don't know if that helped at all, but maybe some of you it helped. So here we're looking at this. We're going to use this little tool to try to do this example. I think I'm just going to change to the circular. That's why I'm having problems. Okay, so I'm going to slide this over. This is the tool we're going to be using. I'm going to put that there. And I'm going to copy some of it. I'm going to clean up some of this. So what we're given in our question is we found this value. This is our Q squared because it's the recessive phenotype. So this value is this number right here. Okay, that's Q squared. So now that we have this Q squared value in place, we can move backwards up to figure out what Q is by taking the square root. And so the square root of 0.09 is 0.3. We know that 0.3 plus whatever value is here has to equal 1. So I can find the difference, and I can say that's 0.7. These values are the same in my structure, so I'm going to put these values, just repeat the values. So this value and this value are always the same because it's always P, always Q. And now I multiply to find the values in the middle. So I'm going to get my calculator out. 0.7 times 0.7, or 0.7 squared, 0.49. 0.7 times 0.3, 0.21, 0.21. These homologous pair here, these two that are exactly the same, those can be added together for our final count. So we have our P squared, our Q squared, and our 2PQ, which is our homozygous, homo-dominant, our homo-recessive, and our heterozygous. And so this value is the value in the top left corner, 49, the value in the bottom right corner, and the value in the middle, 0.42, which is adding these two together. Does anybody like that formula? Does anybody like using a Punnett square? It certainly is easier to do if you've done Punnett squares before, which some of you might have done in a grade 9 course, or if you've done Biology 40S before. If you haven't done them before, you're going to do them a lot later, because our next unit is going to be the genetics unit. Okay. We'll do a couple more questions, and then that will be. Then I'll leave you to watch the video I posted about Darwin for our next section of the unit. So let's do this next one. And this one looks like a nice big question, so maybe we can, maybe we'll do both of these questions. You only need to do enough work on these follow-up questions, on the next generation questions, until you have an answer. Once you have your answer, I don't expect you to keep going and keep solving for more and more problems. Just at that point, you're done. So I think unless it asks you to do extra stuff, but. Okay, I'm just gonna crop this one. Paste another one. Crop it. Short. I'll put that one down there. Okay, we'll come back to that one later. Okay, rabbits can either be short or floppy-eared when the short ears are short ears are dominant over floppy ears. You have 653 individuals, 104 have floppy, 100 and 549 have short. Find the frequency of everything. So step one, frequency of homozygous recessive. Okay, so those would be, so we have to read carefully. Short ears are dominant over floppy ears. So 104 floppy, this is our recessive. And this is going to be Q squared. So let's figure out that frequency right now. 104 divided by, and I need a total. I can either add them together or I can look and see a total right there. So 653. Take this total and divide it away. It does not have to come out to a nice number. It might, but it doesn't have to. So that's 0.1. Looks like I'm going to go with 3 significant figures here. So 159. We're not big on sig figs in our bio class yet, but it is certainly something you'll need to worry about in a university level bio class. Okay, so I have some values there. This is our frequency of, this is our Q squared value, frequency of the recessive allele. Step 2, or that's frequency of the recessive phenotype. What is the frequency of recessive allele? So to get that, I'm going to take my Q squared. I'm going to take the square root of Q squared to find Q. Square root 0.159 equals, if you have a calculator that can do this kind of thing, that's great. I'm going to use some brackets, for example, and I'm going to have to do something like this. Okay, so 0.4 essentially. I guess I should have rounded to 16, and then it would be more obvious. But oh, it's 399. I guess if I'm sticking with 3 significant figures, I can stick with 3 significant figures. 399. So that's my number. This is now frequency of allele. Okay, so that's Q. So now 3, find frequency of dominant allele. So we're going to take 1 minus 0.399, and that's going to give me 1 minus 0.399 equals 0.601. So that's my frequency of the dominant allele. Now that we have the frequency of the dominant allele, we can figure out all the other ones. If you want, you can do Find the rest. If you remember all the parts of the equation, then that's great. If you don't, you could do this square if you like. We can put the 0.3 or 0.6. Might as well just call it 60 and 0.40 up here, 0.60, 0.40, something like that. I'm just going to do some movement here to kind of get everything out of the way. Fine. So this is 0.36, 0.24, 0.24, 0.16. So we have our values. We have, and you could assemble everything in a nice table. That would be a really great idea. So 0.6, 0.4, p squared, 0.36, 2pq, 0.48, that's a combination of those, and q squared, 0.16. That would work if you can label them properly. Say that this is homozygous recessive, heterozygous. We can go like H recess, heterozygous, and H dominant. And then this is the dominant allele. And this is recessive allele. And those would also be good choices as well. And easy for me to understand and for me to award marks for and all that stuff. All right, so the next generation, that's it for that question, though. That's all we need for the question is this end result. Next generation has 560 individuals with short ears and 840 with floppy ears. What's the population? So this is going to be step one, same step as before. I'm taking my floppy ears, 840, divided by my full population. So I have to add the two together. So that's 14. So that's 1,400. So 840 divided by 1,400. And I get a 0.6. And this is supposed to be my floppy ears, right? So that's actually my q squared. So then number 2 is to find q. So I'm going to take the square root of 0.6 to find q. 0. I don't know. Square root of 0.6 is 0.77. And that's going to be my q. And then number 3 is my p. So I'm going to take 1 minus 0.77 to get 0.33. And now I go and think about my equilibrium. Is my system at equilibrium? No way. Look at that. That's a huge difference. I've halved my population's frequency for the dominant allele. So we would say equilibrium was broken. P went from 0.6 to 0.33. And so this would be a point where biologists would start to sound the alarms. They'd start looking for reasons. What's going on? To me, it sounds like it's a farm, and maybe they said, hey, we're hungry one day. Let's eat the short-eared rabbits, and let's save the floppy ones because they're cute, and we can sell them. So all of a sudden, the population has started to move towards that. So that would be my guess, but we'll see. Anyways, that's some ideas. I'm going to leave you with the next half hour to watch that half an hour documentary. I don't think we're not going to watch it together, I don't think, but we do have a classroom activity that I've posted for it. It's just a very simple activity here So make sure you can pull that up. If you have the app on your phone, then at the bottom of your phone, there's a little arrow. Click on that arrow and you'll see your template popped out. So making a theory, who was Charles Darwin? Please give me as much detail as you can. Who was Wallace? Had the other part of that theory. He stands out to me so much that I forgot his first name when I was making the sheet. And then what are 2 observations Darwin made on his trip that contributed to his theory of natural selection? And how do they help formulate these ideas? This is going to be the major part. I really want number three to be the major question. And then in your own words, what is natural selection? And I'm going to put up a couple of other videos, maybe attached to the same worksheet that will let you explore that topic as well. So please take the next half an hour that would normally be class time to explore that movie or that half an hour documentary and any other videos that come off of that you can use that time. All right. So thank you very much for your attendance today. We will be continuing these live streams next week. So same time, same everything. If you need to see me this week, I'm still going to be in the building today and tomorrow. And I will be around in room 208 is where I am right now. So that's probably where I'll stay most of the day. You can see my excitement in this room is that we are currently printing out a chimpanzee skull. So when you get back to school, you're going to have a nice, fun chimpanzee skull to play with. It's printing upside down right now, but it's very exciting. And so hopefully that'll be lots of fun to play with when we get back to school. This unit, how are we doing on this unit? We are at page, just before we wrap everything up, we're at exactly the halfway point. So we are at page 30-ish out of 60-ish. So we are really getting actually quite close. The last half of the booklet will go pretty quickly. And so I don't know if we'll get into genetics while we're away, but I will start posting the genetics booklet pretty soon. When we come back after spring break, it would have been my goal to have this unit done before spring break. We're going a little bit slower than that because of the shutdown, but that'll be the concept. We'll either to finish up this unit this week, or next week, or and start genetics from a mobile online format first. We'll see what happens, and then we'll probably end up doing the assessment for evolution in class or by sort of essay-based questions online. Okay, best of luck. Let me know if you have any questions. You can always send me messages through the classroom. I'll post what can I post here? You can always drop comments on any assignment and I will see them. You can also, I believe you guys can edit the stream if you need to drop a note in the stream. Let me just check. Students can only comment. Okay, I will make it so that students can post and comment. And yeah, that'll work. So yeah, if you need to, you can post directly into the stream now if you need to get a hold of me. Okay, best luck, and we'll talk to you soon.